All right, thank you all very much. It's a pleasure to be here today to talk about some work that we've been doing related to detecting microplastics. Oopsie, wrong way. So microplastics are different from PFAS in that they are unregulated. So there's no regulation for how much microplastics can be present in water. But we're increasingly detecting microplastics in different environmental systems, including in surface water, soils, and also in drinking water. So microplastics are just defined operationally as plastic particles that are smaller than 5 millimeters. They exist in the environment in various morphologies. So you can find fibers, beads, fragments, etc. Microplastics are either going to come from one of two sources. Either they were produced to be at that micro size. A lot of that was done in personal care products, which were actually phased out in 2015. But sometimes you'll find those small plastic beads that were in toothpaste or facial scrubs. But most microplastics we find are probably secondary microplastics, meaning they're coming from weathering of larger plastics in the environment. Fibers that we're finding are probably coming largely from things like washing, synthetic fibers, and that water going into a wastewater treatment plant and then out into the environment. So microplastics are going to come into wastewater treatment plants from all of the various activities where we use plastics, and most of them are removed into the solids, which are then processed in a lot of locations into biosolids. And so our study was looking at could microplastics from originating in biosolids be a source of microplastics then into the environment. So we conducted a study where we looked at agricultural runoff from land applied biosolids, and we also looked at manure to see if we could detect microplastics. We did this at a plot scale at the Rogers farm, the UNL research facility, and we set up 6 plots with replicates where we either applied biosolids at an agronomic rate, a control plot which just had nothing applied to it, or cattle manure. And we just collected rainfall after naturally occurring rainfall events that occurred during the growing season. And so what we found, we did find microplastics both in the biosolids and in the manure, which wasn't particularly surprising. We found a concentration of about 9 microplastic particles per gram of the dry biosolids material. Most of those were either fragments or fibers in terms of the morphology. And then in that corner, you can see the actual composition of those plastics. So the larger sizes, we were able to actually determine the chemical composition and found that they were largely polyethylene terephthalate, but you can see the variety of plastics that were in those biosolids. So I'll show you the runoff data in just a moment, but in terms of potential for movement, vertical movement in soil, at least at this location, we did not see vertical transport. So we measured the microplastic or the soil concentration of microplastics at the top layer and then at a deeper layer at the end of the study period. And we really didn't see much vertical transport, which is good news in terms of microplastics potentially having less ability at this site to transport towards groundwater. We did see microplastics in runoff. And so here's data from our 5 runoff events showing the precipitation and then also just showing the microplastics concentrations we detected in the control. So we still did find microplastics in our control plots as well as in the biosolids amended plots and the manure amended plots with a statistically significant increase in the concentration from those plots that had biosolids. So our concentration range for the biosolids was 16 to 31 particles per liter of runoff that we detected. We didn't detect any microplastics in the rainfall. So if you're wondering where those microplastics came from in the control plots, we didn't find them from rainfall. So possibly not wet deposition, possibly dry deposition or transport by wind, or there could just be some not naturally occurring, but microplastics out there from prior agricultural activities at the site. So we took our data on microplastics concentrations in runoff, and then we tried to estimate for that cropland that we that we use biosolids on for growing corn, how much potential microplastics could be released into surface waters from runoff. So of course, this is going to match the areas of higher microplastics release are going to match where we're growing more of that particular crop. So this would change if we had biosolids application for different types of crop production. But you can see in Nebraska, Iowa, Minnesota, Illinois, quite high potential levels for biosolids to be released from corn production where we're using biosolids as a soil amendment. And then just kind of looking at across the state level, again, and our point here was just to show that we have a lot of areas in the state where we have intensive agricultural production and also a lot of surface water, right? So there's a potential for this runoff then to impact our surface waters across the state. And so if you're interested in more information about this particular study, here's the paper that we published last year. Then the other thing I was going to talk about quickly was just a little bit about microplastics in agricultural watersheds. And so there's been a lot of studies on microplastics in urban and suburban surface water, but we were interested in looking at microplastics in agricultural watersheds as well. So this was just a monitoring study where we collected samples from 4 predominantly ag watersheds. So the Elkhorn, Salt Creek, Johnson Creek, and Big Blue were the water bodies that we investigated. And so these watersheds, as you can see, are largely cultivated crops. We collected 4-liter grab samples from the center of the water body, and then we also collected sediment. In terms of the processing for microplastics, there's a density, a size separation, density separation, and then these were quantified using microscopy. The final step then is characterization of the microplastics using FTIR, which we haven't done yet for these samples. So just showing some of the data for the water concentrations. We did find microplastics at all of our locations with, for the most part, a greater percentage of fibers compared to fragments. The highest concentration, as you can see, was in the wastewater outfall, which isn't terribly surprising, but these other sites across the other watersheds weren't wastewater influenced, and we still were finding the presence of microplastics. These are the sediment data. So also sediment is kind of a reservoir or more like a integrative sampler, right? So you'll probably have microplastics collecting in sediment over longer time periods, and they'll be in the water column. And so again, we saw microplastics present in all of the watersheds in the sediment, now with a higher concentration of fragments, which probably just relates to the size and the ability of those fragments to settle out from the water column. In terms of the size distribution, they were fairly equally distributed amongst our various size classes, although the largest percentage was in our very, very small microplastics, which were 63 microns and below. And then in terms of the density distribution, you can see the amount that settled in DI water versus the sodium chloride and zinc bromide, and those just have increasingly higher densities than water. So the greatest fraction were in our highest density solution. So just to kind of conclude, we did find microplastics in Nebraska waterways at concentrations up to 40 microplastics per liter. We found the highest concentrations at wastewater treatment plant outfalls, but they were also present upstream of the wastewater treatment plant, indicating they're probably coming from more agricultural sources in addition to that municipal wastewater source. We didn't see, I didn't mention this, but we didn't see a lot of correlation between microplastic concentrations and other more conventional water quality parameters. And so this is something that we're continuing to work on, including characterizing those microplastics and trying to look for sources beyond biosolids concentration so we can understand where these microplastics are coming from in ag systems. With that, I'll just thank my students who were involved in this project and funding. And with that, I will turn it over to the next speaker.